All right. Good morning, everybody. Glad you could join today for our trial run webinar for the Technical Assistance Provider Training. I thought it was a good time. I'm Virginia Lamb with NOFA New Jersey, and I thought it was a good time to do a little bit on soils because, of course, soils is the underpinning of everything that we're doing. And we have been to a few locations now, and we've seen some variations with soils. And we, in the initial questionnaire, people said that they felt like they knew a decent amount about soils, but they also felt like they really wanted to learn more about soils. And, I love talking about soils. So we assembled our little group here to do a little overview today. So I'm gonna do just a sort of an introduction 101 soils. Paige is then gonna talk about soil physical, chemical, and biological properties. And then Rajani is gonna take us through looking at soil samples in solution under a microscope and seeing what we look for and what it can tell us about soil health. So why should we care about soils? I always like to start with this because we really rely on soils for everything. Soil scientists sometimes call soils the skin of the earth, and it's like this thin layer that's responsible for all life, and our food, our shelter, fiber, water, and medicine, everything comes from soils. We've really depleted our soils through industrial agriculture and development. But the good news is that we really have the potential to restore our soils to serve many vital functions, which we are sorely in need of, and including plant productivity, nutrient cycling, which includes reducing chemical usage, drawing down and storing carbon, increasing biodiversity, cleaning our water. And these things are collectively known as ecosystem services. And there's been a movement in the past few years to try and put a value on these things, like an economic value, because typically things like this have not been valued economically at all. So big picture, there are 12 soil orders globally. And those soil orders, you can see the names listed. This is just an NRCS. You can just search this chart. Those soils are named depending upon their characteristics. And their characteristics are determined by the formative factors. And soil scientists call these, affectionately, they call them CLORPT, which stands for climate, organisms, relief, parent material, and time. So basically, where a soil is, what's underneath it, how long it's been there, and what factors are interacting on it create the soil. And so when you think about globally, you think about certain regions for certain foods, you think about France for wines, and you think about the breadbasket of the Midwest. Like, these soils are suitable for these crops. So soils can be altered, but there are certain inherent properties that make soil suitable for certain things across the globe. And in general, the composition of soils is like 50% mineral matter and 50% air and water, and then fluctuating amount of organic matter. And then, of course, air and water fluctuates depending upon wetting and drying cycles. So the mineral matter, of course, is the solids, and the air and water is the pore space. Like, if a soil is saturated, there's no air pore space in there. So it's a dynamic system. Soil texture is the percent sand, silt, and clay. I mean, I know a lot of these things are probably familiar to you, but I just like to put it together in this big kind of package. But percentage of sand, silt, and clay. So sand is the big particles in soil, the 0.05 to 0.2 millimeters, the coarse grains. Silt is the intermediary, 0.002 to 0.05 millimeters. And then clay is the very fine particles, very tiny, tiny particles, 0.002 millimeters or less. And the combination of those three things makes your soil texture. And soil texture is not a changeable characteristic. There's nothing you can do to change it. But soil aggregation, soil structure is changeable, and by practice. So when we do, we reduce tillage, we increase organic matter, we change the aggregation or the clumping of the soil, and that affects how the soil functions. It creates spaces for organisms, it creates water channels. So we want to encourage soil aggregation and improve soil structure. And soil health indicators, now I'm going to really not talk about these much at all because Paige is going to talk about them. But the three soil health indicators, they come in under three categories: chemical indicators, which are the minerals and the pH; the physical indicators, which I mentioned, texture, aggregate stability, which relates to water holding capacity, compaction, and so forth; and then the biological indicators. And those can be measured by microbial protein, respiration, organic matter, and carbon. But Paige is going to go into detail on all of those. But I like to take it back to the periodic table because, you know, we do a lot of like casual talking about chemical elements and stuff, but people are a little bit afraid, I find. I do a lot of public education and people are like, yeah, periodic table. So I just like kind of demystify the periodic table, and I call it the ingredient list of the universe. So everything in the world is made out of everything here. And in fact, 95% of everything is made out of like 6 of these things: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And these things are so elements are measured in atoms. That's the smallest unit of measurement. And then atoms combine to make molecules. Molecules combine to make compounds. And then compounds are everything. And then these things are constantly changing depending upon what they interact with. They're turning into liquids. They're turning into gases. They're turning into solids all the time. All these fluctuations going on all the time. So like when I was a kid in school, and I learned science really badly in elementary school, and the nuns in my school would say, oh, your desk is made of atoms really tightly packed together. I was like, okay, sure they are. But it's true, it is. Just familiarly, you know, just to give a visual for what these compounds look like, you know, we talk about them all the time. We even call water H2O, right? The big fat oxygen and the 2 hydrogens. Ammonium nitrate is a common synthetic fertilizer. So it's 1 N, 4 H's, and then another N and 3 oxygens. And then these on the right are like glucose in the bottom. That's a, you know, plant sugar that's made through photosynthesis. And then there are structural molecules like cellulose and lignin. And you can see that all the carbons in there. I want you to hold carbon in your mind for a minute. And see all the C's all through. And even on the lignin, where there's nothing, where there's a blank, that's a carbon. That's how, like how a structural molecule is drawn. Carbon is just assumed. So carbon is this ubiquitous element, right? It's everywhere. It's part of everything. We even use that language. We talk about carbon-based life forms and the carbon backbone of a molecule. And carbon, like most elements, can be in a solid form with carbon that's diamonds or coal or graphite under time or pressure. It can be a solid compound like glucose. It can be a liquid like carbonic acid, sorry. It can be a gas like carbon dioxide. So it changes forms all the time and it's everywhere. And then fossil fuels and some greenhouse gases like methane and carbon dioxide are really largely made up of carbon. So how that looks in the world is everything cycles. Minerals all cycle all the time, right? So the carbon cycle, there's a lot of carbon stored in, you can see this is in petagrams, which is 1 billion metric tons. These are the quantities here. So a lot of carbon stored in the earth, soils, in the crust of the earth, in fossil fuels. A lot of carbon stored in the ocean, a lot of carbon stored in plants. So the blues are called the sinks. That's where the carbon is stored. And the reds are called the sources. That's where the carbon emanates from. So you can see that the fossil fuels were a sink, but then we burn them and they become a source, right? So carbon is constantly cycling like that. So soils have a role. As we degrade soils, it creates respiration, and then the carbon goes up into the atmosphere as CO2. Right. So what we want to do is create situations that draw carbon back down and store it in the soil. So, you know, there's a lot of organizations that talk about, oh, like we don't even have to stop emissions. Soils can be the savior. Soils can get everything right for climate change. And I think soils have massive potential, but I don't think we need to just abandon every other thing. I think we still need to reduce emissions. But carbon sequestration, that's long-term storage, is called sequestration. So that potential is affected by where the soils are, the temperature, the precipitation, the climate where the soils are. So cool, moist soils, like in the Northeast, can hold more carbon than tropical soils. Soil properties, including clay and organic matter content, that matters, how much carbon a soil can hold. Sandy soils can't really hold a lot of carbon. The minerals in the soil affect the carbon storage as well. And then the strategies, the management strategies, and what we do determines whether carbon stays in soil or gets drawn down into soil or gets released from soil. So, you know, I went back to school a few years ago, and because I was so depressed by climate change, but then I found out, oh, soils can really help climate change. So I was really intrigued, how in the world does carbon like get from the air into down into the ground? And it's really two ways. Like carbon dioxide can, it's some mineral oxides. Carbon dioxide can interact with calcium oxides and become calcium carbonates. And then carbon dioxide can interact with magnesium oxides, become magnesium carbonates. But mostly it's via plants. Plants can send carbon into their tissues and in the soil through photosynthesis. And then all these decomposers in the soil can maintain and keep, store carbon, transform it into other compounds, and feed themselves and others. And so this is like my favorite chemical equation is photosynthesis. And it literally, the word photosynthesis literally means to make from light. And I think that's so beautiful. So plants, green plants, have this unique ability to take CO2 and water, and with the energy of the sun, transform it into to glucose and oxygen. It's amazing. But plants will create like a third too much, a third more than they need. Like some they store in their tissues, and some they store in their roots and their fruits and their leaves and everything. But they make more than they need. And I know you soil folks know that they're giving it away. They're strategic barterers, right? They can, so a plant can make its own carbon, but a plant, you didn't see nitrogen in that equation. You didn't see phosphorus or potassium. Like it can't make all its micronutrients, but all these soil organisms can. So a plant gives away glucose, and in return, all these fungal and bacterial communities, insects, transform other nutrients and provide that back to the plant. So it's a beautiful system, and we've interrupted it a lot. And we've disrupted the ability of plants and plant communities to do that, provide that function for themselves. So we can work at restoring that. So I take umbrage when agriculture is like demonized as the climate villain, because it's just another industry, right? It can be better, but certainly it's not the demon that it's made out to be. But agriculture can certainly sequester all of its own emissions, and maybe even more. So that's pretty important, and that's what we're after here. And the 5 ways that you manage soils regeneratively, again, I know this is probably not new for you, is by keeping soil covered with plant residues, by limiting disturbance, by keeping living roots in the ground as much as possible, by creating diverse ecosystems in mimicking of nature, and by integrating livestock. And then also by reducing and eliminating synthetic inputs, which really damage soil, and incorporating organic amendments such as compost and biochar. So just quickly, each of those, we talk about, leaving plant residues on the soil. We talk about adding mulches. It can be any kind of organic matter. I know plastic mulches have been popular, but they're kind of coming under scrutiny. So I did not keep plastic mulches in here, and they don't contribute organic matter. On the left there, I was excited that we had a tour at Salte Farm down in South Jersey, Cohansey Meadows. So I just mulched my tomatoes in Salte, and I was super excited about that. Sent the farmer John a picture of that. So anyway, adding organic matter to the soil, keeping the soil covered on top like a blanket, provides us great safe haven for organisms to do their work. Benefits of living soil cover, roots in the ground. You're constantly adding organic matter, carbon, and nutrients. You're reducing erosion by creating that security and permanence. You're reducing compaction because the roots are pushing through the ground. You're suppressing weeds and pests. In flowering ground covers, you are providing beauty and attracting pollinators and feeding pollinators. And if they're legumes like beans, they can fix nitrogen from the atmosphere. And and most plants have this ability to foster mycorrhizal fungi, which has another benefit. So legumes, a certain class of plants, right, peas and so forth, alfalfa, beans, they have this ability, this unique symbiosis, where they can create these little houses, these nodules on their roots, and then they can recruit these rhizobia bacteria from the soil. And rhizobia bacteria can then transform nitrogen gas, which is 78% of our air, into plant available nitrate and ammonium. It's a miracle. But if you put down nitrogen fertilizer, this doesn't happen. It interrupts the whole process. Arbuscular mycorrhizal fungi called AMF. Most plants have this ability. It's a symbiosis between the fungus and the root. And they produce a substance called glomulose, which I only just learned 2 days ago on LinkedIn. It used to be called glomalin, and it was thought to be a glycoprotein, a sugary protein. But now it's in fact understood to be glomulose, which is a carbohydrate. But it still is the same in helping create stable soil aggregates, that clumping that we want, and provide structure and carbon storage. And then the mycorrhizal fungi are it's like a super highway for nutrients, especially phosphorus and for water, and massive amounts of carbon storage in there. So we really want to encourage those relationships. And the only way we can is by keeping roots in the ground. And if we're constantly tilling, we're constantly destroying that relationship. So we want to minimize disturbance. We want to eliminate or modify tillage, strip tillage, zone tillage. There's a lot of ways you can just cut down on tillage, which is great. We want to diversify cropping systems, utilizing multi-species, doing rotations, and where possible, incorporating trees for the deep root storage. And integrating livestock where possible, but a lot of people don't want to integrate livestock. And so there is a sort of a segment of people that talk about the microorganisms as livestock. They call themselves microbe farmers, and they think about their tiny, tiny little livestock, which that's where I am, because I don't have any livestock. And then again, add organic matter and just decrease chemicals. Always beneficial. And then there's, of course, a great relationship between the healthy soil microbiome and our healthy gut microbiome. And these folks do some really interesting work on that. Just drawing that perfect parallel between soil health, plant health, and gut health. So just in summary, many great potential for significant improvements to soil. Carbon storage is really a low-cost, low-tech way to address climate. And, there's not a silver bullet. There's not carbon sequestration, whatever they call it, carbon capture and storage, all these things that are really expensive and have minimal results, perhaps. There's a lot of different ways we have to address these problems. And again, soil health equals human health. And then just a couple of my favorites. I really love the Soil Science Society of America. You don't have to be a soil scientist to join. They have a lot of great resources. I love the Roots So Deep documentary series, which was featured at this past year's winter conference. Soils for Climate is a great group up in Vermont doing globally a lot of work on restoring soils in agriculture. The Bible book for soils is The Nature and Property of Soils by Brady and Weil. And then I have to say, I'm not on social media. I'm not a social media person, but I do love LinkedIn, and I do learn a lot of great soil science on LinkedIn. Just want to put that little plug in there. So I'm going to stop my share. What time is it? It's 9:19. Okay. I don't know if we want to do questions. I think we should just go ahead though and charge ahead with Paige. Paige, if you're ready.
Yeah, can you guys hear me okay?
Mm-hmm.
I just want to apologize. My voice is a bit scratchy today, getting over some stuff, but
all the
we should be able to
celebrations.
make it. Of course. Yes.
That's great.
All right, let me get my screen set up here. All right, how's that look for you guys?
Looks good. Could you just expand it,
Cool.
to the full screen view? Yeah,
Sure.
Great.
Awesome. All right, so as Virginia mentioned, I'm going to be going over the 3 properties of soil with soil health research and how that really happens in labs and what researchers are looking at based on those 3 properties. So the 3 principles of soil health, they're going to be the chemical, physical, and biological. And these are just some basic properties I'll be going over today. There's more for each of them, of course, but this is just some of the basics that we do. So chemical is often pH, inorganic nitrogen concentrations and content, and elemental analyses. Physical deals with aggregate stability, bulk density, water holding capacity, and water infiltration. And biological principles is everything biological. So microbial biomass, microbial respiration activities, organic carbon and nitrogen contents as well. So jumping right into chemical, pH, as we all know, affects a lot of nutrient availability and cycling in soils. This is mostly done through affecting enzymes and how specific nutrients can be made available. This is mostly measured using a pH probe in the lab, which is pictured here on the right. I find that the stand is extremely helpful when you have a lot of soils. The inorganic nitrogen is usually measured in what's known as a calorimetric assay. So inorganic nitrogen is nitrate and nitrites and ammonium as well. So this is a specific form of nitrogen that is available for plants to uptake through the soil. Organic nitrogen is something completely different and is not available for plants. So getting this specific measure is really helpful to know what nitrogen in your soil is actually available for your plants, what can be taken up by that. So calorimetric assays work by combining different chemicals and putting them into a plate reader. So it measures how different the color changes, and the more it changes, the more of the nitrogen is present. And different elemental analyses, there's a lot of different methods for these because, of course, as Virginia mentioned, we have the whole periodic table happening. So it just measures the concentration of other elements, including trace metals, micronutrients, and heavy metals. These can be beneficial for the soils or they could hurt the soil. So it's really important to be able to measure how much of these you can have. Now, touching on the physical aspects, there's a lot of physical aspects of the soil, but just the three we're talking about today are aggregates, bulk density, and water holding capacity. So aggregate size and stability, sort of as Virginia mentioned earlier, aggregates are clumps in the soil that are held together through things like organic matter and roots from the plants. And they really affect the physical structure. And they do this through pore space and pore connectivity. Larger aggregates have a lot larger spaces and more pores connected between them. And these spaces can be hotspots for microbial activity, and they can fill with water or fill with air depending on the saturation content. And they're really important because they also deal with compaction. The more pore space you have, the less compaction. But as Virginia mentioned, with the management affecting pore spaces, more tillage, more management destroys these aggregates, makes them smaller, and it really disconnects the pore spaces and can really minimize the pore space, which then in turn negatively impacts our microbial interactions. So these can be measured both dry and wet measurements using a sieve shaker, which is pictured on the right. You put your soils in on the top and it'll shake it and separate all of those sieves or have different size holes in them. So it'll separate the sizes of the aggregates. And you can use the wet measurements by running water through there as well. And that's important to know how weather and erosion is really affecting your soil. So bulk density measures the weight of a given volume of soil. So this can really give you an insight into what percent of your soil is pore space versus what is physical mineral matter. And more pore space, as we mentioned before, deals with microbial interactions, but also water infiltration. So if you have more connected pore space, the water is going to be able to get deeper down into your soils as opposed to just running off the top. And water holding capacity is the water in the soil that is available for plant uptake. It's in those pore spaces. It can be reached by roots. And the way we measure this in the lab is you compare the dry soil weight with a saturated soil weight, and you should be able to calculate what percent is actually the water that is available in those pore spaces. And then for biological, there's a lot of different methods you can do for all of these traits, but just touching on a few again, we'll start with microbial biomass. Microbial biomass is sort of a difficult thing to measure. You need a lot of machines for biological methods. What we do here is we compare the soils with and without living microbial biomass. So essentially, you compare after extractions, you just have the total carbon content of your soil, and you compare it with the total carbon content of a soil that has microbes that have been killed. So by using that and machine and different calculations, you can actually find out what is the weight of the actual biomass of the microbes. Similarly, microbial respiration can be measured as well. And this is more looking at, as opposed to how many of the microbes there are, what the microbes are actually doing. So with respiration and activity, what is the carbon dioxide that they are producing? So the best thing to do here is microbial respiration. You can perform an incubation with your soil. So we often just put them in jars and are able to extract gas from that. So we use a tracer, stable isotopes, carbon-13, so we can actually tell what is the carbon-13, what is the carbon that they are taking up and turning into the carbon dioxide gas. So that by getting a measure of that, we can understand how quickly are they respirating, how active are the microbes in the soil. And then for organic carbon and nitrogen, these are measures of soil organic matter. We're all familiar, I'm sure, with the organic matter in the soil and how important it is for both the soil life and the plants that we grow in it. So using these, similarly to microbial biomass, we use this specific TOC machine to do this analysis. And it's the same extraction process as microbial biomass, so they can all be done together. But what we're measuring here is really the total organic carbon, organic nitrogen that is available for microbes to uptake. So I talked about inorganic nitrogen earlier. This is the separate organic nitrogen that's not available for plants, but it's available for microbes. And then moving on to enzyme activity, we're really just looking at what are the enzymes that the microbes are using in this soil. And there's a lot of enzymes and a lot of microbial activity that happens. So there's a lot of different processes you can do to use this, but mainly we use fluorometric assays, which involves adding chemicals that will fluoresce when the enzyme is active. So once it breaks a molecule, it fluoresces and the plate reader can see, oh, how much of these molecules are being broken by this enzyme. And that gives an insight into microbial activity, such akin to microbial respiration as well. And then lastly, identification of your soil fauna through microscopy and through sequencing is something that's super important, but can either be really easy if it's large macrofauna or very difficult if there are lots of microbes or you have to use sequencing. 16S rRNA is the best sequencing method that we typically use. It's faster, it's cheaper, and you can tell which species of microbe you have in your soil. And then just this picture here in the bottom right is from my sophomore, I want to say, year in the lab. This is during our microbial biomass extraction, where you combine the soils with K2SO4 and extract them out before sending them to the machine. And then with our chemical, physical, and biological indicators, that is all the stuff we can do in the lab, but how can we actually put these into the field? It's the most important thing to see what is accessible for farmers to do in the field. But unfortunately, a lot of these tests are really difficult to do. Some of the things we can do in the field are pH for chemical analysis with the portable pH probe, which is pictured in the green on the right. For physical, you can measure water infiltration and holding capacity, which is pictured in the bottom right. So what happens there is you have cylinders that are driven into the soil. You add water and then you time how long it takes for the water to drain into the soil. And then through different calculations, you can measure the actual water infiltration rate and holding capacity. You can also bring portable scales. That's pretty easy to do into the field, and that'll help you measure things like bulk density as well. But unfortunately, biologically, there's not a lot of field options because the microbes are so small, of course. We can't really see them. You can identify macrofauna like earthworms, beetles, other larvae. But in terms of microbial processes, you need a lot of smaller machines to be able to do this. And I think that being able to advance the biological tests that we can do in the field is going to be really important. There's a lot of cutting edge research on this that is currently happening, and the way I think that we'll be able to make this more accessible for farmers is really focusing on how can we get this done in the field to really understand the biology you have in your field. Because doing it with your hands on, is a lot better perspective than sending it to a lab and having them do it and send you a piece of paper back. So with that, I just have some references here, but biological field wise, I want to pass it over to Raj and she can talk about what we did at our samples.
That was excellent, Paige. Thank you.
Thank you, Paige. Can I start? Okay.
Yes.
So this is a demonstration. I don't have any slides. Let me turn on the video. Hi. So we're going to go over the rules to collect the soil sample. These are the soil samples we collected on 4th of June from IB Farm and Comeback Farm. You label them with the date and time. Can you guys see this? Okay, cool. And then they are kind of anaerobic. It's like 2 weeks from now, right? So I'm not going to test these guys. I took a sample from my backyard, and that's what we're going to use. This is 1 milligram of backyard soil we'll be testing today. And this is the distilled water. We are going to do 1 to 10 ratio. So I'm going to put up to 10 mark here. And then shake it gently for 30 seconds. This is 10 milliliters. And then we're going to shake it. While I do this, here is the microscope. I think everybody is familiar with this. Can everybody see this? Okay. This is the stage. These are the lenses. I have 10, 4, 40, and then 100. And this is the BioVid camera, eyepiece, which we are not going to use. This is the control that the stage goes back and forth and sideways. This is the coarse, I don't know if you guys can see this, coarse focus and then fine focus. That's the major parts of, and this is the epifluorescence. We'll be using this one today. I have a root from the hazelnut tree that I got from Foggy Bottom Farm. I forgot the name of the farm. It's in South Jersey. I got a hazelnut tree. It looks like shrub right now because when I try to pull this root, it looked like it's growing some side branches. Maybe it's not a tree. Maybe it's just a shrub. But I got this, and then we'll be looking at this root for mycorrhizae today under epifluorescence. So here are the slides. Slide and the cover slip. This is almost ready. And we're taking 1 drop with this pipette. I'm going to look under the microscope. Initially, I'll be looking at the slide with 10 objective lens, which is 10 multiplied by another 10 that's built in. So it's going to be 100 magnification. And we'll look through the slide in a pattern for nematodes. That is called nematode scan. And can I share my screen?
Yes.
Okay. Yeah, this is the software that I use to look at the organisms. Yeah. And while looking at it, this is the software I use to record everything. This is from the Soil Food Web school that I went to. I entered the details first, where I got the sample, timing, and everything in this screen. Once I have all this put in, now it's going to give me a problem because I didn't put in. Next thing I do is nematode scan. That's what I just talked about, where we go over the slide with 100 magnification. And then I do main assessment, where I look for fungi, actinobacteria, and other organisms like protozoa, meaning testate amoeba, flagellates, ciliates, everything. And then lastly, I do the bacterial count. Once I do all this, I go here, and then there is a result page that I can bring up, which will show fungal to bacterial ratio and all the information that a farmer can use to build his soil. So this is the software. I'm going to close this. It's going to give me, yeah. Back to this. So back to biological assessment. So I'll let it stabilize for like 10 seconds. And then I take just below the organic matter, I'm going to take a drop from here. And I'll let some drops go. And put this on the slide. And then cover it with the cover slip. Second. It's not sure. This is epiflorescence, and then we are in the okay. So we are at 100 magnification. And then I'm adjusting the condenser, bringing it up, and then everything is in focus. I'm using the fine focus to, yes. So this, can everybody see the screen with the microscope?
Yes.
Okay. So I'm going to go to one edge of the slide and then start from there to look at the entire slide. This is one edge. So I stop here and then start going sideways. And I go in a pattern to look for nematodes. The weather is wet, so I'm hoping to see some nematodes. Usually they are there when the soil is wet. We can find them easily. I see some big aggregates. That looks like some kind of a plant material, like cellulose. Stop me if you have any questions. Yeah, right there. So this is a nematode. And we're going to look at it with 400 magnification so we can take a better look. The problem with these guys is they won't stay stable. They always move. The way I check if it's not calming down is I just take fire and just go near it for a few seconds, and it will startle, the fire will startle the thing and it'll stay stable. But based on the mouth part, this is very aggressive, I can't tell right now. But based on the mouth parts and the body parts, morphology, I can tell what kind of nematode it is. I'm sorry, it's not staying stable. So in the interest of time, I'm just, I can. No, too aggressive. In the interest of time, I'm just going to go. That's how you do nematode scan. So you basically go over the entire slide and then note down how many nematodes and what kind they are. Mostly they are beneficial. Occasionally you'll find some root feeders. So after the nematode scan, you will go over the slide, looking for other organisms. Hopefully you can see the bacteria. The It is these small guys, they are blinking, I mean, not blinking. You can see them, they are wiggling and moving a little bit. I don't know if is it visible? Can somebody confirm?
I can't see any wiggling. Is that what you're asking?
I brightened it just now a bit. Does it
I
help?
see some movement.
Yeah.
do?
These guys, they are cockeye. And then,
Oh, yeah, I see now.
yeah. So when I go up and down like this, you should be able to see them wiggling. Those are all bacteria.
Oh.
And this is testate amoeba. There is an opening here, and this is the
Mm.
test of the amoeba, and that's good. And there's something moving here. Based on the
Mhm.
movement, it looks like a flagellate. I need to see it properly to see the flagellum and really decide what it is.
Oh, look at all that movement.
Yeah, it's going a little fast. It's gone. But
He's,
that's,
be-
you
he's
get the
behind
idea,
that.
right?
He's behind that big piece of carbon over there.
Yeah, I think so. So it went somewhere. So these are cysts. And then this is what do you call that? Spores, fungal spores. And this is another testate amoeba. You know this because there is food inside and the mouth is like this, like, flat on one side. I see a lot of organisms. I'm trying to look for, yeah, here is another beautiful testate amoeba, a lot of food inside. And there's something wiggling, but it's not coming out, but that's okay. And these are the things I note down in the other screen of the software. A lot of testate amoeba. I'm looking for fungal pieces. I thought I had some fungal rich. So yeah, here. This went bad a little bit here. Maybe I shook it rather aggressively, but this is a fungal hyphae. I know this because it's different from the background color and it has septa and it has uniform diameter throughout. And usually the darker color ones are good. So this is good fungal piece that's extending almost couple of fields of view, which is good. So I will note down all this in the other screen and then go for bacterial count. There is no way I can do a bacterial count in this screen. So I dilute this solution more. And after a couple of dilutions, I should be able to count the bacteria. So this is about biological assessment using the microscope. And Virginia talked about mycorrhizae, forming symbiotic relationship with the roots. So if you don't have any questions, we'll move to the other slide where I have a piece of root that I just picked from, oh, you can't see me. I'll share again, but this is the root I picked and then I placed it on the slide and I'll share again. I'm going to put this on the stage right now and then turn on the epifluorescence. I'm going to go back to 100 magnification and then turn off the and then turn on the epifluorescence. And we'll be looking at the root. Give me a minute while I bring it into focus. Yeah. So this is the root. And this is just the root. And then to see the insides of the root, you need to really focus and see this pattern here. The yellowish green glow says that there is phosphorus present in the root. And then this H kind of pattern within the, one second, maybe I can do a better job. In the root, yeah, this pattern right here says there is mycorrhizae present in my roots. I think the darker pieces of indication of some kind of iron. Yeah, this is the pattern that you will look for to make sure you have actually mycorrhizae colonizing the root. I think this is the best example I can focus so far. Yeah.
So is that the endomycorrhizal? Like, is that the inside the root?
Yes,
Include
yes.
the ecto? Nice.
No, endo is inside. Ecto is.
Oh, sorry. I always
Yeah.
mix those up. Endo, inside, right.
Yeah. So yeah, actually this is better here. This is the inside of the root. This is the root. This is just the root with the hairs on it. And then you will see the inside now. This is actually good. Yeah. And this is the pattern you look for to make sure you have not that well defined, but it's like the septa is hidden inside and it's just like, it looks like H. This root actually looks good. I don't think this is damage. This is just some kind of, because I didn't clean it. I just brought it from the tree and then I didn't do much with it. I just picked it up. Yeah, that is how you look for what's in the root. Yeah. If there are no questions, I think I'm done.
Awesome. Excellent job, both of you guys. That was great. Exciting news. We just ordered and received a microscope for our
Awesome.
use. Yeah, so we'll have that available for our next getting together. So everybody can have a look and see. And you both can school us in the finer tips of using a microscope and what that tells us.
The OPEX one? Which one did you get?
The one that Dujan has. I think I'll have to find the information and send it to you. And yeah, I think it's exciting. Paige, you mentioned about, that a lot of these are, of course, lab tests, and they take a lot of time and they're expensive, and they don't really give the farmer the hands-on information to understand what the information is being provided. So I think it's great that we can work together to develop some simple field assessments for people. They can do themselves, they can see, they can really understand what to be looking for and what to be promoting. So I'm excited to have the opportunity to do that together. All right, so I think that's it, unless Johan or Scott have anything to add.
Nah, nothing for me.
No, it was a phenomenal presentation all the way around. The microscopy is always fun. It's amazing how large comparatively those nematodes are to the rest of the sample. Like they're big predators of either, sometimes of your plants and sometimes of soil biology. And it's amazing when you look at that and you zoom in, you see like just how much motion there is in that soil. You see that bacterial shake. When you see that from a live soil versus something that's not very alive, it's a huge difference.
Sorry, let me see if maybe you guys, that earbud's not always a great.
Better.
I looked at your soil too, Scott. It looks good.
Yeah. To the others, fantastic going over of soils. You know, from a farmer's perspective, I feel like pH is often a pushed really hard, is super important to watch, and people are always adjusting their pH. I think it's important to keep in mind that your plants need nutrients; they don't need a measurement, and pH is a trailing factor telling you that your nutrients are a little bit out of balance. So, frequently it's telling us that we need calcium, but you need to look at your calcium number to see whether that's the nutrient that is needed to change your pH, right? Now, you could have your pH out of balance and you could have tons of calcium in the soil. You might need to get some other micronutrients in balance. And the only other thing that struck me was in the discussion of nitrogen in testing. There are not many crops where it's worth doing a soluble nitrogen test. I would say corn is one of them. If you're growing sweet corn or field corn and you can respond quickly with some sort of a water soluble nitrogen. I feel like for organic farmers, it's that ENR, that estimated nitrogen release, which basically comes from evaluating how much biology is in your soil, how much organic matter is in your soil, and then being able to calculate how much will natural respiration and biological activity kind of release as nitrates over time. Because, you know, we're not in the nitrates business the same way in organics that they are in conventional ag. You know, we're not putting on a ton of water soluble nitrate and ammonias. So we want to see that biology, that organic matter, which can be seen from your on-farm test with the microscope, can be seen through your organic matter percentage through the soil lab tests. But yeah, when you have good biology and you have good organic matter, you will have good nitrogen release over time, and that's important. Right? Your plants need nitrogen for their whole life, so having a good high estimated nitrogen release is a good thing to look for in your soil samples. This is a great presentation. I really appreciate you guys putting all this hard work in.
Thanks, Scott. Thanks always for your great input and additions. Really
Absolutely.
appreciate that. Yeah. All right. Do we adjourn?
I
See
second.
y'all. Huh? All right, so that wraps. I think we're a little ahead of schedule, which is great. So we'll be convening at Cotton Cattle Company on Wednesday. Hopefully getting started nice and early and maybe wrap nice and early if it's going to be super hot. And I think that's it.
Thank you.
All right, thank you all.
Thanks a lot.
Thanks, everybody.
All right, take care. Bye. Bye.